Good morning or good afternoon also from my side, and happy to be here today. Nobody sees me. I don't see anyone. I guess it's normal nowadays in that time, but I hope you will still enjoy this talk today. So as Ralf already mentioned, today I'm going to focus about our work around the TCell Bispecific Antibodies platform that Roche is heavily investigating. And also I will talk about some different combinations with checkpoint inhibitors, but also with immunomodulators, mainly focusing on the 4-1BB axis in that particular publication that we recently did. But before we go into that, I just want to quickly give you an introduction about what are T-cell bispecific antibodies, because some of you may not be so familiar with. So Roche is using the T-cell bispecific format as depicted here. Actually, all the TCBs under development, especially ones under clinical development as of now, are using exactly this format. And this format is very important for being, for the T-cell bispecific functionality. So all the TCBs have a bivalent binding to the tumor target, here specifically against CA, which is a tumor antigen expressed in colorectal cancer, but also gastric cancers and others. And it has a monovalent binding site to CD3, human CD3 epsilon, which is inverted. So we call this format inverted. So it looks a bit awkward if you first have never seen it before, because the CD3 doesn't sit outside, but it actually is located inside. So that's what we call an inverted format. But very important is also that the Fc part is silenced. So we don't have any interaction with Fc gamma receptors anymore to prevent any cross-linking via Fc gamma receptors and CD3. So we really want to aim to have a T-cell activation really only in the presence of the tumor antigen. And those TCBs have a long half-life. It's like most of those have an IgG profile when injected into humans, but also in mice, of course. And today I'm going to focus on the CA TCB, but later on I will talk a bit also about our heme-targeted one that targets CD20. But apart from the TCB platform itself, of course, just quickly how we are generating our humanized mice here in-house. So we use basic NSG mice. They're pretty young, between 4 to 5 weeks when we use them. And the first thing we do is we inject busulfan IP with a simple injection. Here we were titrating this out a bit in the very beginning to not have much of an impact on the health of these animals. And we found that 15 milligram per kilogram is actually a pretty good dose that is standard very well or tolerated well by the mice, and also gives very good engraftment later on. And 24 hours later, we inject the CD34 positive cord blood cells IV, so very simple protocol, as you can see here. And around 15 to 20 weeks after the stem cell injection, we do screen all these animals. And at the moment, we are generating around 300 to 320 animals every month that serve then for particular project needs and requests. We are doing flow cytometry to screen all these animals, and here's just one batch just to give you an idea. So these are 300 mice, and you can see that the average engraftment with using that protocol is around 60%. But of course, it's pretty heterogeneous because we are using 10 mice per donor. So we have 30 donors in one humanized mouse batch that later on will then allocate it to different studies. Of course, what we know, and everybody that works with humanized mice is aware of, we have primarily TM B cell engraftment. Earlier, we have some B cells coming up, but later, around 15 weeks, we start having also the human T cell population present. And here's just a snapshot. We did, of course, also some other characterizations. Here's human blood and focusing on the main human immune cell subsets with NK cells, T cells, B cells, and myeloid cells. And when you do the direct comparison to the blood of humanized mice, you already see that the myeloid compartment in that particular strain is not represented as efficient as it could, and the myeloid cells are way reduced compared to the human compartment. But we clearly get a very nice TMB cell engraftment, and also the NK cells in that particular strain is very low in frequency. That's true throughout the different organs and also in blood, of course. But the main advantage why we are heavily investigating in those animals is that the T cells and the B cells are generated de novo in these animals, and so you have kind of tolerance induction of the human T cells and the B cells, which, as you know, when you inject PBMCs into immune deficient mice, you get a strong GVHD, which also limits your window of experimental window, and also doesn't really allow you to do mode of action studies because the T cells are pretty highly activated. So, but now focusing on the two different questions I wanted to talk with you today about is what is the mode of action of those T-cell bispecific antibodies in vivo, and also what are potential escape mechanisms upon TCB treatment and how can we mitigate those. And focus on the first question, so what is the mode of action of TCB? We started with CA-TCB, and this is also the one that I was just talking about before. So this is the one targeting colorectal cancer antigen. Here, what we did, we used humanized mice and we injected MKN45 cells subcutaneously into humanized mice. And this is what you can see here. So this is an image of a humanized mice. We labeled the drug, so CA-TCB is labeled in red, and then an untargeted controlled T-cell bispecific antibody is labeled in green. We call this TP47, so it's just irrelevant binding site. Both of them have been labeled and simultaneously injected into these tumor-bearing humanized mice. And here you see time kinetics. And what you clearly see is already starting at 24 hours, you start seeing this accumulation of specifically the CA-TCB, so the tumor-targeted TCB, in the tumor area. Whereas the untargeted one seems to be still not concentrating in the tumor tissue, but rather being disseminated all around the body of those humanized mice. Also, and that's very important, since of course you have humanized mice used here and human T cells present, the CD3 binding is very low in affinity, but the tumor targeting is very high in affinity. So the CA binder is really high in affinity, and that's why you see preferentially the binding to the tumor site and not much in periphery. And this is also what you can see when you do the analysis. So here, focusing on the CA-TCB, you get a reduction of the signal early on in blood, and in spleen it's rather stable, but you see a nice accumulation of drug over time until 120 hours. Well, this was the last time point we measured here. And for the untargeted TCB, of course, the signal is going down in all of those compartments. So now what we did in the next step, we used this particular cell line, so it's the same one. We injected those into humanized NSG mice, and then we randomized those animals into different treatment arms and started a treatment here with CA-TCB. And here we have given them twice weekly with 2.5 milligram per kilogram and have basically monitored the tumor growth inhibition. And this is what you see here. So CA-TCB as monotherapy is giving some tumor growth inhibition. As you can see here, it's not super striking, but clearly you have an effect of the drug here in humanized mice. And at the same time, and this was the main focus of this particular study, is also the mode of action. So we have done extensive immunopathy analysis by flow in the tumor, but also by cytokine measurement from the tumor lysates. And what we have seen here is that we get a very nice increase of human T cells in the tumor upon CA-TCB treatment here at termination, which is pretty like more than 10 times as much as in the vehicle. And also very important is we get infiltration of both of CD8 and CD4 human T cells, but the shift is clearly on CD8 cytotoxic T cells here, which shifts the ratio, of course, up to 2 or even higher sometimes, depending a bit on the dose used. But more interestingly as well, is when we analyze the phenotype of those T cells infiltrating the tumor up on the TCB injection, we do see clearly a PD-1 upregulation, 4-1BB upregulation, K67 and also granzyme B upregulation, indicating that we also not only increase the frequency of T cells in the tumor, but also through that cross-linking, specifically in the tumor, we are activating those T cells that eventually induce the tumor cell killing. When we do the same analysis in blood, where you don't have any tumor target present, we don't see any of those phenotypical changes on those human T cells in humanized mice. And this is what I mentioned before. We also used some part of those tumors that were explanted at study termination to prepare tumor lysates, and we analyzed different chemokines, but also cytokines. And as you can see here, we get a nice upregulation of interferon gamma and IL-2 TNF, some of the pro-inflammatory cytokines, and these are human cytokines. Also IL-10, and also important to mention here, because I will come back to that later, we also see a nice upregulation of different chemokines like CXL9, 10, and 13. They are all interacting with CXCR3 expressed on memory T cells, and that's an important point that I will later come back. So we verified this exactly those findings also in other models. So we used the pancreatic cell line as well, injected into humanized animals. Very similar as I just showed you. It's also CA positive, as you can see here. So it's more patchy, this expression. The cell line also has more stromal regions as compared to the MKN45. But it's generally lower in terms of CA expression. And then we did the same thing. We injected tumors, randomized, and started the treatment. And also here we see a good tumor growth inhibition, no regression, but a good tumor growth inhibition. And we see the same finding as we just discussed before. We see a nice T-cell infiltration up on the treatment. The ratio is shifted towards CD8. We see form B granzyme upregulation, also PD-1, K67 in that particular model is more or less the same, but we saw pretty similar findings in the second model as compared to the first one. So since we did extensive ex vivo analysis, we were thinking of creating a kind of TCB effector score or TCB treatment score that kind of summarizes all those immunomobility findings and helps us to then track the activity of a TCB in vivo or later on also in patients. And this is basically depicted here. So there is a heat map with all the analysis we did by flow cytometry here on the right, but also on cytokines and chemokines here. And here you have the treatment with CA TCB and vehicle, and this is all tumor data. And here you already see very nicely that you get upregulation of those different readouts on the CA TCB treatment arms, but there are also a few that rather had an impact on the cellular level, but not so much on the cytokine level. And when we looked by PCA, we identified some of those readouts as most significant, and these were then suggested to take into consideration when we created this TCB treatment score. And this is what we try to depict here. So we found that MIB alpha, CXCL10 and 13, but also the general T-cell infiltration, CD8 T-cell infiltration, and PD-1 expressing CD8s especially, all of those seem to be really significantly and describing the TCB activity. So we kind of merged all of those readouts into one. If you're interested, of course, you can go back to the publication. It's depicted also here. So each of these parameters actually got a ranking in the quartile. And then since we had 5 readouts that we identified or as most important, you come up with a maximum number of 15, and then you divide them by 15 again. So of course the details you can look up, but this is a score that we found that nicely describes the TCB activity in the tumor up on the treatment. So we were using this score also for subsequent studies to really make a point on how active the TCB actually have been after the treatment in humanized mice. But most interesting later on was, of course, also we see this nice T-cell infiltration up on treatment. We were wondering what is actually the exact mechanism of this T-cell recruitment. Is it T-cell recruitment only, or is it also intratumoral T-cell proliferation after the TCB treatment? And here my colleague was just recently publishing a very interesting data on that. Here we also use humanized mice, but not in a way I was just talking about, but rather in a way of as a donor mouse, because what we did here was we implanted a skin fold chamber on the back of the skin of normal NSG. So these are not humanized NSG mice, so blank NSG. 2 days before, so 2 days after the surgery of the skin fold chamber, we then injected a lymphoma cell line that was labeled in blue and resident T cells that came from a humanized mouse. And this is very important because these T cells are now tolerant against the NSG background. If you do the same thing, and we did, with just isolated T cells from human PBMC, you have such a strong reaction against the mouse tissue that there is no movement of T cells anymore. So this kind of analysis or experiment would not be possible with T cells that are reacting against the mouse tissue. And here I have to explain, of course, we here injected a lymphoma cell line, so it's not about CA TCB, but the CD20 TCB, but it's exactly the same format. It just targets a tumor antigen on B cells and not on colorectal cells. But the principle is exactly the same. So the therapy was mixed together with this mixture. The CD26, this Moran cell line, is just as a bit of a placeholder to have a bit more separation of the tumor plaques. And then 3 days later, we started doing the imaging. But of course, the most important thing I just forgot, we did not just co-graft those T cells from humanized mice, but we also at the same time injected in a second color labeled T cells, also derived from the same humanized mice, but IV. So these are the T cells in the periphery, and the pink ones are the ones that have been co-grafted. And this is how it looks in the 2-photon. So here you have vehicle, and here then there's the ones that got treated also with CD20 TCB. And you see here the resident T cells, so the ones we co-grafted with, are here in pink. And here the recruited ones, potentially recruited ones, are in yellow. So, and what you see here as a summary is when you had added CD20 TCB to this mixture of recruited T cells and lymphoma cells, only then you get an upregulation of or a higher infiltration of recruited T cells to the tumor site, indicating that CD20 TCB is cross-linking the T cells that are present in the tumor to then further attract T cells from the periphery. And very interesting is as well, then the next study we did, we titrated the T cells that were co-grafted with these lymphoma cells initially, so 0, and then 1 to 100, 1 to 10, or 1 to 1 ratio. And what we have seen is the more T cells are present at baseline, so here, co-grafted, the more cells get attracted to the tumor from the recruited T cells that were ejected IV. But we were, of course, wondering why or how is this mechanism really working. And so what we also knew already from in vitro assays, this is depicted up here, is when you have CD20 TCB treatment in vitro together with T cells and lymphoma cells, you do see very nicely interferon gamma upregulation in the supernatant, and you can also stain this interferon gamma in the CD8 cells. So it's of course secreted by the T cells up on CD20 TCB engagement. And what we have also found in the supernatant is that you get CXCL10 upregulation that you can simply detect when you do these co-cultures with together with CD20 TCB. What we've also seen is that CXCL10 is also upregulated in the presence of CD20 TCB. Here it's simply when you add interferon gamma on the tumor cell. Sorry, I just forgot to mention that. Here is just interferon gamma added to the lymphoma cells, just telling you that CXCL10 is actually secreted by the tumor cells, or potentially can be secreted by the tumor cells. And when you have CD20 TCB in, again, these co-cultures, you see also CXCL10 upregulation. And here's a kinetics of the same co-culture. And you see at 3 days, you get the highest values for the concentration of CXCL10. So what we did then was exactly the same study as just described here. But what we added on top in this mixture is an anti-CXCL10 antibody or an anti-interferon gamma antibody, just to verify if this is the axis that is important for the T cell recruitment. We had another control, which were tumor cells that were pre-treated with interferon gamma and should then secrete CXCL10 and were also implanted in the skin fold chamber. And this is the readout, so the recruitment of these peripheral T cells. And this is extremely interesting because when you see here CD20 TCB, and this I showed before, you get a nice recruitment of the T cells that were injected IV. And when you block CXCL10 and interferon gamma in this mixture, you don't get any of this recruitment, really indicating that the mechanism, most likely the hypothesis is that you need a certain number of T cells present at baseline that will then be cross-linked, interferon gamma will be released, and then this interferon gamma will subsequently induce CXCL10 upregulation to recruit CXCR3 positive memory T cells. We also tried and verified this in vivo. So we did efficacy study with CD20 TCB in lymphoma-bearing humanized mice. So it's the same mouse model. And at the same time, we tried and blocked interferon gamma and CXCL10. As you can see here, CD20 TCB is extremely efficacious and completely regresses very large lymphomas in humanized mice. And the blocking of interferon gamma and CXCL only had a minor effect. I mean, there was an effect, was also significant in some time points, but it wasn't super striking. But also on an immunopathy level, we also saw that you got also some reduction in the immunopathy, so we had less T cell infiltration when we blocked CXCL10 or interferon gamma. I have to mention here that what we did not do is a Gazyva pretreatment, meaning when you inject CD20 TCB here, you have a direct cross-linking of the peripheral B and T cells inducing a CRS in humanized mice, which already a bit makes it difficult to then block interferon gamma and CXCL10 because you have already such a big cytokine storm. So there will be no specific tumor or no CXCL10 or interferon gamma tumor-specific depletion or inhibition. So we would need to repeat that to really verify this also in vivo in humanized mice. So now coming to the second question, which is about the potential escape mechanisms and also how we can mitigate those. I'm going back to the study I was describing to you in the very beginning when we used CA-TCB in humanized mice in the MKN model. What we also did at the end, not only doing flow cytometry but also histology, and we verified that the tumors that got treated with CA-TCB are really also show this increased T-cell infiltration by histology. But what we also looked at was the PD-L1 expression, and we nicely could show that after the treatment with CA-TCB, you get the nice upregulation of PD-L1 on the tumor, which is of course also something we quantified. And here's just the positive area, which is clearly upregulated with when you have CA-TCB injected into humanized mice. We also did bulk RNA sequencing from those tumors that at study termination. And also here with the bulk RNA sequencing, looking at PD-L1 or PD-1, we just see the same thing. So also on an mRNA level, we see PD-L1 upregulation, but also PD-1 upregulation, confirming also the flow and histo data. And generally, we also highlighted here, but you can go for the details also in the publication. We highlighted here the most significant changes as well. And some of those candidates already I was just talking about, and here's a nice verification about CXCL10 and others. Granzyme was everything that we found. It was also upregulated here by bulk RNA sequencing. And here's just some gene ontology references where you can see that most of those signatures are really related to inflammation and T-cell activation that we are inducing with CA-TCB in the tumor in the humanized mice. So of course now, I'm very sorry, it's a spoiler already because I cannot do a presentation mode here. So just don't look at the right-hand side. But we also looked at the T-cell active or the PD-1 and PD-L1 upregulation after TCB treatment in vitro. And you see that whenever you treat with a TCB, you get also nicely PD-L1 upregulation on the tumor cells in vitro as well, here on MCAN45, here on the MFI. And we also see PD-1 and PD-L1 upregulation in vitro on the T cells, as this is true for the CD4s, but also down here for the CD8 cells. And of course, next step is now that we see that this axis seems to be important and can be one reason why we don't have a strong regression eventually in humanized mice here. We, of course, did combination studies, not only with CA-TCB and PD-L1 as monotherapy. So PD-L1, by the way, is not doing anything in that particular model in humanized mice. But when we combined, we saw a pretty decent combination effect. As you can see here, is a first-line treatment. So we started the concomitant injection at the same time from the beginning on. But here we also had an experiment where we had the second-line treatment where we saw that CA-TCB is losing more on its activity, and only then we started this PD-L1 combination. And you can see that from that time on, we induced the stasis of the tumor as compared to just CA-TCB. So this was, of course, this was a combination with checkpoint inhibitors and TCB. So it's one potential escape mechanism, so the PD-L1 and PD-1 axis. So it seems to be something that is important for TCB activity. I have to just also tell you that when we do look in those tumors that have been treated in these combinations, we did not see much of additional immunopathy findings. So granzyme, as I showed before, granzyme is already upregulated, PD-1 is upregulated. So we also couldn't find much more in terms of T cells. So the frequency was also not much increased in those tumors. So it seems to be something related to the functionality itself. But we couldn't really find striking PD markers in humanized mice, at least, that could explain this combination. But we also, and this is talking now about another publication that we recently published, we used the same model as well in the MKN45 bearing humanized mice, and we combined it with a FAB-targeted 4-1BB ligand. And this tumor is actually, when you stain for FAP, you can see really nicely that this tumor also expresses in the stroma nicely the FAP. That's why it made, of course, also sense to have a combination done with FAP4B ligand and 4B. For the ones that may not be so familiar with, 4B is also a costimulatory receptor that is expressed on activated mainly CD8 cells, not so much on CD4 cells. And that we wanted to target because it makes a lot of sense, because we reported that 4B signaling is preventing the activation-induced cell death, and this is something that the TCB, of course, can also potentially induce. And this is the same model. So we have here the combination with CD8-TCB and FAP4B ligand, where you see a very striking combination effect as compared to monotherapy arms in here. And since I mentioned with checkpoint inhibitors, we didn't see much of an additional T-cell infiltration when we added FAP4B ligand here in this MKN45 being humanized mice. You see it is very, very strong additional T-cell infiltration here in the tumor. So my last slide is again about a CD20 TCB and another molecule that is similar to the FAP4B, but it targets CD19. And the bispecific antibodies, the CD20 TCB, as I was referring to before, and we have one particular model used in humanized mice that is depicted here. So this is OCLA18. When you inject this in humanized mice and you start a treatment with CD20 TCB, you see a very nice tumor regression, and the tumors are almost not visible anymore. But then those tumors come back on treatment and escape the treatment. And this was very interesting to us. So we also looked into by flow. We looked into the expression of 4B, and we also see that 4B is upregulated after CD20 TCB in humanized mice in the T cells, so the CD8 cells in the tumor. So we wanted to use CD90 4B ligand that gives again co-stimulation via 4B. On the CD19 positive lymphoma cells. So what we did the same study as here, and we started the second line treatment with CD90 4B ligand, more or less the day when they started to escape. And what you can see here is basically in gray, these are the escapers with just monotherapy of CD20 CB, but when you inject CD90 4B ligand, you are able to keep most of those tumors under total control. And this is just as a time to event analysis. So you clearly see that this is extremely efficacious, this tumor, and that 4B agonism can really prevent also escape up on TCB treatment.